Okay, this is your Invertase Lab video. This lab, what you're going to do is you're going to be determining the optimal hydrogen ion concentration and the optimal sodium ion concentration for the catabolism of sucrose using invertase extract from Saccharomyces cerevisiae. So I'm going to go ahead and break that down right there. Okay, determination of optimal hydrogen ion concentration, which is pH, and sodium ion concentration, which is salt concentration, how salty something or salinity, right? For the catabolism of sucrose, which is the breakdown of table sugar, using invertase extract from Saccharomyces cerevisiae. Obviously, invertase is an enzyme with that suffix. And then Saccharomyces cerevisiae is bread yeast. So you're going to have this lab over multiple days. You're going to be assigned in groups of 3 or 4. Now, the materials available to you, of course, additional materials, like I mentioned later, will, may be available if you request them. If they are not necessarily needed for something, I will, save you the time. But if you have a good idea, I want to hear it. And the reason why I open it up to additional materials is because, again, you are responsible for producing the protocol for this lab and designing this experiment. So some of the materials that I have available for you, of course, is the Saccharomyces cerevisiae, so you'll have the invertase source, some triple beam balance, ruler, graduated cylinders, test tubes, again, of various sizes, salt, which is going to be your cation concentration, so determined for salinity, distilled water, hydrochloric acid, sodium hydroxide. Of course, the sodium hydroxide and the hydrochloric acid are your alkaline and acidic sources when determining your hydrogen ion concentration. Hopefully remember that hydrochloric acid, which is an acid, would have a high concentration of hydrogen ions, while sodium hydroxide, a base, would have low hydrogen ion concentration. Now, if you're wondering how would you be able to tell if there's a hydrogen ion concentration that's high or low in your solution, that brings us to our next material, which is a burette. And for those that forget what a burette is used for titration. So we're going to do a couple titrations. Actually, going to do probably around 8 of them, just so you can get some good averages. You'll have some acidic standards and alkaline standards, so you can do your proper titration. You'll have your 2 acidic samples and 2 alkaline samples. We'll probably do your 2 acidic samples on one day and then your 2 alkaline samples on another, just because that will allow me time to switch out the burettes, because the titration with an acid to a base will require different materials in the burettes. So, and of course, when I go the other way, I'll need different solutions in there. You'll have an Erlenmeyer flask, which you'll use for your titration, and of course, you'll have a pH indicator, probably phenolphthalein, but something along those lines. You'll have a heat source, a Benedict's testing solution, and sucrose and glucose. Now, the reason why we have the heat source and the Benedict's testing solution is because the Benedict's test is what we're going to use to determine if the invertase actually broke apart the sucrose. So sucrose is going to be it's a disaccharide, and it's composed of a fructose and a glucose monosaccharide. Well, as it turns out, when a Benedict's solution is added to a solution that contains glucose and it is heated up, it will turn bright orange. Now, if it's a negative result, then obviously you don't have a change, in which most of the time that Benedict's test solution being blue, your solution will remain blue. But with the Saccharomyces cerevisiae sampling, it's going to be kind of cloudy. So you guys are going to have to try to manipulate how to gain your positive and negative controls. Now, with the sucrose, obviously we're going to be using that to test the invertase as our substrate, but I did include glucose in your materials, so you can derive some sort of positive and or negative control. So some relevant formulas or equations that you need to know. You need to know what the dilution formula is. Okay, you guys can look that up online. Pretty much looking at volume and molarity and how one affects the other. And then you'll also need to refresh your memory on balanced chemical equations. As it turns out, hydrochloric acid and sodium hydroxide is a fairly easy one, so it shouldn't take you that long to freshen up on that particular neutralization equation. Okay, so if you guys have any questions, by all means ask. Again, you're responsible for trying to put this thing together, okay, and I'll direct you as best I can. But don't count on me giving you any direct answers, because I won't. Okay? You guys have a good day, and I will see you tomorrow. If you have any questions, shoot me an email.